travaille sur le planaire. Alors c'est atypique encore comme séminaire, mais l'année dernière, vous vous rappelez, on avait déjà eu un petit séminaire sur les planaires. L'idée c'est que nous, dans le laboratoire, on essaie de développer un modèle d'hôte interaction pathogène, donc c'est toujours bien que quelqu'un qui vienne nous expliquer ce que c'est le planaire, puisqu'on s'en sert pour ce modèle. Donc il va nous parler cette fois-ci de régénération et il va vous montrer pas mal de mots, on va dire, le planaire avec plusieurs textes et autres. Voilà, donc Johan, présente your data. You're welcome.
Thanks, Eric, for the introduction, of which I didn't understand the words, so I hope you
But I'm sure that they understand at least one part of the talk, which was mine.
I just understood planaire, but I hope that's going to be okay. Okay, so yeah, thanks very much for the invitation. It's a real pleasure being here and seeing what you guys are all up to. So basically what my lab is interested in, or what I think has been my driving force to actually become a biologist, is the diversity of animal body plans that are out there in nature. So you find things with wings and things with fins and then things with lots of little bristles sticking out from the side. And of course all of these things look different and all of these things look recognizably different, so that just by looking at an animal we can tell which species it belongs to usually. And they all do that simply because they all have unique genomes. And somehow the genomes determine the shape and the proportions of the organism. But I think how exactly this is happening, how a genome molds the developing tissue masses into the species-specific shape of the organism is still a really big question. And this is exactly the huge number of questions that my lab is interested in and that we are trying to address. Now we try to address these questions relating about the conversion from genome into shape using these animals here. And now if we wouldn't have our favorite MAC-PC conversion issues, we wouldn't be able to see the Planarians, our model system, crawling through this film here. But since I will have to do my presentation about movies, we move on right away to the next picture. And so here, this is the model system that we are studying. This is a flatworm of the species Schmittea mediterranea. Actually, they probably are pretty common around here as well. And these animals are about, well, 2 to 3 centimeters long when they are fully grown. As the name flatworm suggests, they are rather flat in body shape. And on first sight, they look like a rather misguided choice of model system when trying to study the shape of an organism, simply because they don't look very exciting from the outside. But what these animals can do is actually the following. So in this case, I have cut this worm into 18 different pieces. So once down the midline, and then 8 times across. And of course, with just about any model system out there, this would be the end of any interesting biology, and you would be left with a bloody, boring mess in your dish. But what happens in planarians is that each one of these little fragments here will rapidly seal again, and this happens by the epithelium on the dorsal and the ventral side of this tissue piece flattening out and crawling over the wound edge, sort of like fibroblasts. And what you see here, this white stuff spilling out, this is in fact the mesenchyme, the soft internal tissue of the animal. And then within about 2 days, you can see the appearance of this whitish tissue here along the wound edges. And this is in fact the so-called Regeneration Blastema, which is a common phenomenon associated with regeneration just about the entire animal kingdom. And the Regeneration Blastema is essentially a very fascinating type of tissue that can, in a context-dependent manner, regenerate and rebuild whatever has been removed by the injury. And in planarians, this process is very fast, so that already after 2 days, you can start to see the appearance of eyes on one side of the blastema, but on the other side of the animal not. So essentially by 4 days, this animal has already specified here's going to be the new head and there's going to be the new tail. And then within about 2 weeks, each one of these fragments up here will have reshaped itself into a perfectly proportioned little miniature planarian. And so essentially I think the great uniqueness of the Planarian model system is to give us a way to experimentally determine the starting point of body plan morphogenesis, and this of course is a very unique asset in trying to studying the mechanisms that actually determine the shape of an animal. Now the fact that Planarian regeneration works so well is all the more astonishing given that there are actually rather complex animals. So when you look at the anatomy of planarians, you essentially find a typical complement of animal organ systems. So planarians have a rather well-developed brain that actually contains most of the neurotransmitter systems we also find in other animals. So for example, they have dopaminergic neurons, serotonergic neurons, and GABAergic neurons down here. Then in addition to the brain, the planarians have a very well-developed digestive system. And actually the digestive system consists of epithelial tubes that branch into finer and finer branches that invade every last corner of the body plan. And this probably is a mechanism of distributing the nutrients within the planarian body plan, because the one organ system they are missing is a circulatory system. They don't have a heart and they don't have blood vessels. So obviously this is an asset in regeneration if you can't bleed to death. And then third, planarians also have these a very well-developed expiratory system, a so-called protonephridial expiratory system that we'll talk a little bit more about today. They have as a peculiarity only a single body opening. And this body opening is adorned by the so-called pharynx, which is basically a long muscular tube that they use for ingesting food and for ingesting waste products. And then finally, planarians are also literally full of this very specific kind of stem cells, which actually only this year have been shown to be a pluripotent stem cell that is able of generating all the different cell lineages in this rather complex animal. And then again, just getting back to the point, all of this complexity, all of these cell types, in their proper orientation and organization, can be regenerated from any random piece like this, cut out from anywhere in the planarian body plan. Now, of course, the big question is, how can this happen? How can a random piece of tissue reshape itself into a complete animal? And on first sight, one can simply make two very, very, very basic assumptions. And the first one is, we clearly need some sort of signaling systems that instill order on a local basis. So basically by local, I mean cues that would organize the differentiation and the agglomeration of cells forming an organ, for example. And secondly, there also needs to be a global patterning system that specifies, for example, here be the head, here be the tail, this be the dorsal surface, underneath be the ventral surface. And during my talk today, I'll just tell you two little stories relating first to a local patterning system that we've started to work on, and secondly, to the more global patterning problems of how to specify head and tail identity in planarians. Now, about the local systems, in order to study local cues that, for example, shape organ morphogenesis, we decided to have a closer look at the planarian expiratory system. So now, here to the left is basically a cartoon of the planarian expiratory system, as you would find it in textbooks since the 1950s. And this drawing here is essentially based on early light microscopic observations at the turn of the last century, so without any markers or any fancy biology whatsoever. And basically what these early researchers found, together with electron microscopists in the 1960s, was that the planarian expiratory system consists of some sort of tubular structures, which are adorned on their end by these fascinating cell types called flame cells. And basically each flame cell forms this complicated cap structure here that sits at the end of a tubule. And the flame cell contains on the inside of this cap structure, it projects a thick bundle of cilia. These are about 200 cilia that point downwards towards the opening of the tubule. And what the concerted beating of these cilia does is to draw tissue fluid from the surrounding tissues across these very fine filtration slits here into the tubule. So essentially, the flame cell is a single-celled ultrafiltration device. And then once the fluid has been pumped into the inside of the tubule, it is presumed to be traveling down the tubule and to be modified much like the human kidney would deal with its ultrafiltrate. And now here we found a way of staining actually the planarian expiratory system simply by using antibodies against tubulin, and this works really well since most of the excretory system is densely ciliated, so there's lots of tubuli in there, which is why we see it very nicely with this primitive staining method. And what you see here, these fine dots spread all around the surface of the animal, these are, in each one of these dots is actually a single flame cell. And when you zoom in on such a region here, you see that each flame cell is connecting to a thin tubule, to a thin ciliated tubule, and several of these tubules are then converging on a common structure that is slightly thicker. And at some point we lose the structure, presumably because it transitions into a part of the tubule that's no longer ciliated, so that we lose it. Now, what we wanted to study in particular, looking at the Planarian excretory system, was this question of organogenesis during regeneration. And in principle, we were interested in this very basic problem of how do you rebuild an organ from starting from such a random piece of the Planarian body. And of course, also this level here of staining is not enough to actually say something about how this organ would form. Therefore, we needed cell type specific markers. And screening a whole bunch of in situ patterns, or a library of in situ patterns, we finally arrived at this set of 3 markers, which are here shown in multicolor in situ hybridization, overlaid on this alpha-tubulin staining in blue that you've seen in the previous slide. And what we found here is this one gene called inexin 10. This is a gap junction protein that labels the peripheral branches of the protonephridia all the way up to the flame cells. And then in addition, we found a carbonic anhydrase that connects the ciliated part of the protonephridial system, or always connects 2 neighboring ciliated parts of the protonephridial system, and actually marks a part of this tubular system that is no longer ciliated. And then in addition, we found an axonymodynine, so basically which makes sense, being expressed specifically in the ciliated flame cells. And so using these 2 markers and high pressure freezing electron microscopy that we also developed recently, we can now arrive at the following description of the Planarian protonephridial system. So essentially, as we already saw in the alpha-tubulin staining, we are dealing with a highly branched epithelial organ that at its tips has the flame cells labeled by alpha-tubulin because of the cilia and the axonymodynine. Now the flame cells connect to tubular branches that are positive for the inexin 10, positive for the inexin 10 gap junction gene, and these what we call the proximal segments of the tubule, they connect to the more distal segments of the tubule that are positive for the carbonic anhydrase. And also in electron microscopy pictures, you can actually appreciate that the morphology of these different cell types is very different and highly complex. So for example, this here is a cross-section through a flame cell showing the bundle of cilia on the inside. And you can see out here these fine filopodia that make up the filtration diaphragm that draws the tissue, that filters the fluid while it is passing inside the flame cell lumen. And then, for example, the cells of the distal part of the protonephridial branches, they have a very smooth inside membrane, but a highly convoluted outside cell membrane. So practically, these cells are trying to maximize their surface to volume ratio, which is commonly seen in absorptive cell types. Now, having these cell type specific markers available, we could address the question of whether the protonephridial system regenerates de novo. So you could imagine new structures just arising from scratch in a blastema, or whether in fact this organ would regenerate by growth and extension from existing protonephridial remnants in the tissue. And in order to get at these questions, we did a time course experiment with our 3 markers plus alpha-tubulin. And what I'm showing you here is always only 1/2 of an anterior blastema here and here, or 1/2 of a posterior blastema down here and in this panel. And I'm only showing half of the blastema simply because the exact same thing is happening on the other half of the blastema in perfect mirror symmetry. And what we start to see, actually, and I'm just summarizing this down here, is that very early on we see this scattered expression of our proximal marker. We don't know if this is real or if it's just background. But the first structure that we see reproducibly and invariably, always in perfect mirror symmetry on the left and the right blastema half, is what we call the inexin 10 positive prototubule. And by electron microscopy, we know actually that this structure is initially a solid rod of cells that then gradually, over the next couple of days, starts to develop a lumen. And interestingly, just if you concentrate on the panel here on top, the entire complexity of the protonephridial system seems to, in fact, originate from this prototubule structure. So on day 3 of regeneration, we always find the subdivision of the prototubule by now the first carbonic anhydrase positive cells. And already on day 3, we start to see branching and branch extension from the structure. Now by day 4, this branching and branch extension continues already towards the periphery. And by day 6, essentially it seems as if the complexity of the protonephridial system in the new tissues has been regenerated completely. Now we spend a lot of time trying to find connections between this initial prototubule and possible protonephridial remnants down here in the old tissue. But we couldn't find anything neither by electron microscopy nor by serial section confocal microscopy. So we think that in actual fact protonephridia regenerate de novo in the blastema, so that this blastema essentially would have capabilities similar to embryonic tissues in which cells would condense and form new organs. And then secondly, the origin of this prototubule is now one of the bigger cell biological questions that we are trying to address. So whether, as I just mentioned, whether this prototubule forms by cell condensation or whether actually also invagination from the surface epithelium might be involved. And in addition to these more specialized questions, we are of course also interested in the actual molecular mechanisms that participate in shaping such a complex branching morphogenesis process. And now, in order to find molecules in planarians, we at the moment have available the following two main tools. So first, we have rather robust Rnai screening. We can do basically organism-wide knockdown of specific genes. This we can do relatively simply by expressing double-stranded RNA in bacteria, pelleting the bacteria, and simply mixing these bacterial pellets with liver, which happens to be the favorite food of planarians in the lab. And then once the worms have ingested this bacteria-liver mixture, we achieve organism-wide knockdown of genes. For example, shown here in the case of beta-catenin, that's universally expressed all across the animal, and after knockdown there are only tiny little remnants remaining at the very tail tip. Secondly, the genome of the model species Schmittea mediterranea has been sequenced, and also my own lab has been generating a lot of transcriptome resources now, such that we have a pretty good idea of the gene complement in this organism. And putting these two things together, RNAi screening and knowledge of the genome, of course the fastest way of arriving at molecules involved in a particular process is to carry out a candidate gene approach. And what we did in this case was to clone a library of about, by now, around 400 or 500 signaling or genes, all components of conserved signaling pathways. And wherever possible, we try to clone both the extracellular ligands and the transmembrane receptors and the intracellular transduction machinery in order to have a comprehensive overview over pathway function. And now the question was, okay, fine, so we have a way of knocking down all these genes that may be involved in protonephridial regeneration, but how can we actually recognize a protonephridial phenotype when we see it in an RNAi experiment? And here we noticed that when we knock down either this inexin 10 gene, of which we knew that it is specifically expressed in protonephridia, or when we target components of basically microtubule-based ciliobiogenesis, then in many, many cases we ended up with these strangely transparent animals that were massively expanded. And moreover, we know that this expansion, this massive expansion, is actually due to massive tissue edema formation. So essentially, whenever protonephridial components are targeted, or when cilia, which are of course also heavily enriched in protonephridia, when cilia are no longer functional, the animals accumulate huge amounts of water in their tissue, such that in cross-sections you hardly find any cells anymore, and most of it is just empty water-filled space. So first of all, these results showed that protonephridia in planarians are in fact required for osmoregulation. But secondly, they also gave us a nice little tool for identifying potential protonephridial phenotypes in a simple screen. And one of the molecules we pulled out from the screen was now a Planarian EGF receptor. And I should say that planarians, quite unusually for invertebrates, have 5 different EGF receptors. So usually invertebrates only have one EGF receptor. And one of these EGF receptors seem to have a specific role in protonephridia biogenesis or protonephridia function, as indicated by this massive edema formation, both in intact animals or after regeneration of amputated RNAi animals. And just very briefly, we are perfectly sure that this is in fact an EGF receptor because it blasts to the human EGF receptor, for example, with extremely high E values, and also the domain structure is clearly that of an EGF receptor. Now, given the role of EGF receptor, or well, of tyrosine kinase receptors in general, the first question now was whether this protein has a specific role inside protonephridia or maybe more general physiological functions. And in fact, the expression pattern of this gene is already highly protonephridial specific. When we look now again in multicolor in situ hybridizations, we in fact find this EGF receptor to be rather specifically expressed in flame cells, together with our existing flame cell marker. And moreover, during regeneration, we also find that the EGF receptor becomes expressed right around the same time as our other flame cell marker. So therefore, the conclusion is that EGF receptor 5 is in fact specifically expressed in flame cells. And now, given that EGF receptors generally can have functions on cell differentiation, or in other cases, again, the wider family of tyrosine kinase receptors can also influence morphogenesis events, the question was which one of these would apply in the case of the edema formation defects that we saw when this EGF receptor was in fact knocked down. And so in a first pass, we simply examined all our other markers. Here, just as a quick demonstration that, again, RNAi is working very efficiently in planarians. So when we target the EGF receptor itself, we basically lose all detectable expression of the EGF receptor with this rather crude assay, I admit. But most of the other cell type markers seem to be there, except for the flame cells. So here, if you look closely, you can see in the bottom panel that even though the strong pharynx expression remains, what disappears are in fact these very small dots in the periphery, which in fact correspond to the flame cells. And moreover, this disappearance happens relatively rapidly after the end of RNAi feeding, where you can see a gradual decrease of the number of flame cells per protonephridial unit, down to about nothing at the end when the animals are just about to explode and die. So therefore, clearly, the EGF receptor is required for the maintenance of flame cells. But what we noticed, in addition, was that during regeneration, in absence of this EGF receptor, there were also massive morphogenesis defects. And in fact, what we found was that the early regeneration was relatively normal, but as you can see, between day 3 and day 4, now things start to look very different from control. So whereas in control animals we saw a lot of branching and branch extension, in the case of EGF receptor 5 RNAi animals, the branches just remained as thick little bundles that also additionally projected ectopically towards the posterior. And now, in fact, when we let such animals go for longer and now look at the final stage of protonephridia after about 2 weeks of regeneration, we find that especially in the tail, things are going horribly wrong, and protonephridia turn into these coils of tubule segments with very, very few flame cells associated with them. So basically, in addition, we have these defects. And so in conclusion, what our analysis of this EGF receptor showed in planarians was a combination of cell specification or maintenance defects, and at the same time associated morphogenesis defects. And in fact, this set of phenotypes was very reminiscent of the of phenotypes one can observe in other branching morphogenesis organ systems, such as, for example, the Drosophila tracheal system or the developing human kidney. And in both cases, the elaborate branching structure of the organ is actually established by specifying specific cell fates at the very tip of the developing branches. And these specific cells are highly motile and actually actively pull the developing tubule branches out into the tissue environment. And so the hypothesis we are pursuing at the moment in our system is that in planarians, or during the branching morphogenesis of the protonephridial branches, flame cells may have exactly the same role, namely as a tip motor that draw out the branches into the developing tissue or into the regenerating tissue. And now a second aspect of this protonephridial story that you may have noticed already is that at least the topological organization of these invertebrate protonephridia is highly reminiscent of the vertebrate kidney, or at least functionally reminiscent of the vertebrate kidney. So what happens in the functional units of the vertebrate kidney is that in this little capsule here, the Bowman's capsule, blood vessels form these massive coils in which actually blood fluid is leaking out into the lumen enclosed by the capsule. And this fluid, actually, this ultrafiltrated fluid, is then passing into the tubule where it is progressively modified during its passage before it is finally excreted as urine. So now the functional arrangement of these two structures looks very similar. Of course, in the one case, it is blood pressure that is actually driving the ultrafiltration. And in the case of planarians, it is ciliary beating that is driving the ultrafiltration. But so whenever such a resemblance between two structures occurs now in evolutionary terms, of course the question is this convergent evolution based on functional constraints, or is it in fact evolutionary homology between these two structures? And in order to test that, and simply also just out of curiosity, we now started to look at a number of kidney-specific genes in humans and started to see where we find them in where we find their planarian homologs on protonephridia. And one of the first genes we looked at is this molecule called nephrin, which in fact is a human disease gene. And nephrin is a major component of the filtration diaphragm that is required for keeping big proteins in the blood and preventing their loss in the urine. And we find this protein in planarians exactly at the corresponding site, namely at the filtration diaphragm of the planarian flame cell. And I have to say that we also have, in the meantime, we have mapped many more additional molecules. So at least some molecular components seem to be the same. But recently, the group of Peter Redin in Boston has in fact carried out a transcription factor analysis of the transcription factors required for specifying planarian protonephridial cell fates. And what Peter's group found, in fact, is a range of transcription factors that are required for the specification of protonephridial cell fates. And these transcription factors, just when you look at this list, are basically a who is who in vertebrate kidney cell fate specification. So I think these two results together therefore argue very strongly for evolutionary homology between the between the invertebrate protonephridial excretory systems and the vertebrate metanephridial excretory system. And now basically what this means in the end is that the excretory system evolved only once before all these lineages split, but the more practical conclusion from this result is that if this is really turning out to be true, and we are testing this at the moment, then also the planarian protonephridials might in fact in the long run be a good model system, a good invertebrate model system for human kidney disease, because as you might know, so far only the invertebrate model systems like Drosophila or C. elegans, they have very derived expiratory systems that don't combine ultrafiltration with fluid processing any longer. So, now basically this was just a little excursion into the short-range cues that govern organogenesis or organ morphogenesis in planarians. And for the remainder of the talk, I would like to concentrate now on the global positioning system, which must exist in order for the animal, for such a little piece, to now start regenerating a new head or start regenerating a tail in the proper place. And in search of these molecules, we again utilize the same signaling library, simply because when you look at early animal development, then you will find a highly conserved number of signaling pathways that shape a Drosophila embryo in the same way as they do a vertebrate embryo, or maybe, in this case, also Lophotropisone embryo like Planarians. And so we are dealing with a conserved set of pathways, signaling pathways, that across evolution pattern body plans. And now in order to figure out which ones of these or which one of the molecules specifically are required for Planarian body plan patterning, we again relied on Rnai Screening, this library, but this time using specific amputation paradigms to, for example, ask in the first case whether particular molecules are required for head and tail regeneration, or by simply chopping off a piece from the side of the animal, we can screen for things specifically required for mediolateral regeneration, or by simply looking at animals or observing animals after they have eaten RNAi food, we can then also start to look for things that are required for maintaining the shape of the organism once it has been established. And one of the pathways we looked at first was the winged beta-catenin signaling pathway. And you probably know this pathway inside out because it crops up everywhere, no matter if you are studying development or if you are studying cancer or human pathology. And basically, this pathway consists of secreted extracellular proteins, the so-called winged ligands, which bind and recognize frizzled transmembrane receptors. This is a family of seven transmembrane receptors. And then, once the these receptors elicit a complex intracellular signal transduction cascade, which culminates in beta-catenin. So beta-catenin, in a way, is the business end of the pathway. And in the absence of ligand, beta-catenin is constitutively degraded by this destruction complex, keeping it essentially out of the nucleus and preventing it from turning on gene transcription. So now, in order to simulate the off state of this pathway in planarians, we simply targeted beta-catenin and knocked down beta-catenin, and then asked whether we would see any anterior-posterior patterning defects. And in fact, we did. So sorry, this was also meant to be a movie, so it's just a still frame, but it doesn't matter. Because what you can see that in these animals in which beta-catenin is no longer present, the animals are perfectly fine in terms of regenerating a head. The only problem is that they no longer make a tail. And instead of regenerating a tail, they just put another head there in place of a tail. And in this case now, from this picture, you might simply think that it is only two eyes appearing in otherwise tail tissue. But when you look carefully, and we've done that really exhaustively, then by all intents and purposes, these are really bona fide heads that contain a brain, that contain all specific gut branching, and so on and so forth. And moreover, these ectopic heads are perfectly functional. And if the movie were playing, you could see that the two heads in this poor animal are actually constantly engaged in a tug of war, where both of the heads are trying to crawl into opposite directions. So clearly, therefore, the conclusion from this very simple experiment was that decreased beta-catenin activity is in fact necessary and sufficient for head specification in planarians. Now, of course, we also wanted to do the opposite experiment and ask, okay, what happens now if there's too much beta-catenin in the system? And since we still don't have transgenesis in planarians, we had to do it indirectly. And the way we did it was by now targeting this negative destruction complex. And basically by removing the destruction complex now, at steady state, beta-catenin can no longer be degraded, so therefore its level should go up in the cell. And the experiment that we found, or the result that we found in this case, was almost too simple to be true. But what happens in such animals is now that they are perfectly able to make a tail, as they should, but now instead of making a head, they make another tail. And so in this case, again, we are dealing with exactly the same situation. This is a bona fide tail. It has all the components of a normal tail, and it is perfectly functional. And so in this case, actually, instead of pulling away from each other, both of these tails push towards the center of the animal continuously. So what this result therefore showed was that increased beta-catenin activity is both necessary and sufficient for specifying tail in planarians. So, and what these results together then show is that in fact the planarian decision of whether to regenerate a head or a tail revolves around the switch-like properties of beta-catenin, where increasing beta-catenin signaling means now regenerate a tail, and decreasing beta-catenin signaling means make a head in this position. And the question now, of course, becomes what are the molecules that regulate the switch? And that this is of vital interest to the animal is actually illustrated quite graphically by the following picture. So in this case, we are looking at an animal, and an intact animal that has not been cut, so it's not regenerating in any way. And this animal has been fed with beta-catenin, RNAi food. And as you can see, after about 2 to 3 weeks, this animal starts to form new heads all around its perimeter. And in fact, even the tail is converted into a new head. So therefore, the decision to regulate this beta-catenin switch is really one that is very, very crucial to the animal. And now, in trying to, so basically, having defined this machinery now, it is clear that when we cut an animal somewhere, then at one side, beta-catenin has to become upregulated, and on the other side, beta-catenin has to become downregulated. Now, when we look at the components of these signaling systems, at the other components of the signaling systems that specify head or tail in planarians, then in fact we find that things that normally activate beta-catenin signaling, so for example the Wnt ligands and so on and so forth, they tend to be all expressed in the tail of the animal, whereas things that on the other hand inhibit beta-catenin signaling tend to be expressed in the head of the animal. So this makes sense, right? Because head means low levels of beta-catenin signaling, tail means high levels of beta-catenin signaling. But now imagine that we would cut, for example, we would cut the animal right here in the middle. So in the middle, basically, very few of the tail specification genes are expressed, and very few of the head specification genes are expressed. So therefore, clearly, all of these components that are required for specifying head versus tail identity need to be made or need to be expressed de novo at the wound site. And this proposes now a little problem here. So somehow these systems have to become reactivated depending on the context of the wound. And the question now is, how is this happening? Are these molecules, once you cut out, for example, now a tiny little fragment from the center of the animal, as I've graphed it down here, will it just randomly somewhere around its perimeter activate, upregulate beta-catenin on one side to make the tail and downregulate it on the other side to make a head? Or is there in fact a memory of the polarity in the original animal that then also triggers the switch in accordance of the original polarity in the animal where this fragment came from? And actually, and this is also the beauty of the Planarian system, that you can do very, very simple experiments to test such rather profound questions. And for example, in this case, we are cutting unilateral triangles from the head region of the animal. So in both cases, the triangles are identical, except that in the second case, the point of the triangle is pointing forward, and in the first case, it is the flat side of the triangle that's pointing forward. And what we find now, inevitably, is that in the first case, in the first type of triangles, the eyes are always appearing on the long side of the triangle during regeneration, whereas in the second case, the eyes are now always appearing between the points of the triangle as down here. So what this very simple experiment therefore shows is that clearly there must be some sort of memory of the original polarity in each and every Planarian cell, ultimately, in order to allow the cell then, if it so should happen that it is localized at a wound, it can then decide that here should be a head, or if its other side is exposed, that here should be a tail. And now, interestingly, and this was relatively recent work from the lab of Nestor Oviedo, so they managed to also generate double-headed animals now with a drug, so in this case actually gap junction inhibitor. And what they could do with this experiment, where they can simply wash out the drug after it has done the damage and has induced ectopic heads in this case, what they can do then is to cut these areas again and ask whether the formation of this new head here has now left permanent effects on the tissue polarity in this animal. So in other words, during subsequent regeneration rounds, would this animal keep making new heads here, or would it maybe revert back to making tails as it should have in the first place? And in fact, what they found in these experiments was that regeneration in turn dictates polarity. So basically meaning that once you have made a new head, or once cells have triggered this beta-catenin switch and decided to become head, then this also repolarizes permanently the adjacent tissues. So basically what we are left with is a circular self-organizing system in which pre-existing polarities specifies regeneration polarity, but then regeneration polarity again specifies the tissue polarity for subsequent rounds of regeneration. And in fact, just as my last slide, I want to give you our current model of how we think Planarian regeneration can be so extraordinarily self-organizing. And what we think, and I didn't have time to show you all the data that goes into this model, but what our current model assumes that there are two fundamental cues that planarians need for successful regeneration. The first one is a molecular difference between the dorsal and the ventral surface of the epithelium. The second one is the still mysterious intrinsic tissue polarity that we were just discussing. And what we think happens first in regeneration is that when the wound is sealed by the migration of the dorsal and ventral epithelium over the wound edge, that where dorsal and ventral cells meet around the wound perimeter, a specific edge organizing center is established. And we think that it is in fact this edge organizing center which is then capable of interpreting the intrinsic tissue polarity, such that if the vector of the intrinsic polarity hits it at right angles, this wound edge will decide to set up a head organizer, in this case meaning to down-regulate beta-catenin activity, and in the second case, where it would just generate up-regulate beta-catenin activity in order to generate a tail. So basically, and this effect in itself then would set the stage for the next round of regeneration, because these organizers forming at the end of the fragment here would in turn reinforce, or in case something goes wrong, repolarize the existing tissue polarity. So therefore, I think in general, regeneration is intrinsically a highly self-organizing problem. So because in contrast to embryology or embryogenesis, embryonic development, where you can put, for example, head determinants in a specific place and tail determinants in another place in order to initiate head or tail formation, in regeneration you can never rely on such pre-localized determinants, simply because each and every part of the organism must be able to make both head or tail, simply depending on the chaotic nature of injuries and depending on the exact exposition of the intrinsic polarity cues. So therefore, because regeneration in this sense is a rather self-organizing system, we are now trying in my lab in Dresden, we are trying to address regeneration, in fact, as a systems biology problem, using a lot of computational and transcriptomics tools in order to get at the molecules and understand how they organize in the tissue environment. And this is where we are at. This is Dresden at its best, so to speak. I would like to thank the people in my lab and also people in my former lab in the States, in the lab of Alejandro Sanchez Alvarado, where I did some of the work I presented today, and especially Han Tikin Wu, who did most of the protonephridial data that I showed. And this was our institute just about 2 weeks ago. So you can see we have harsh winters in Dresden, but nevertheless it's a beautiful city. So if any one of you would be interested in maybe doing a postdoc in this regeneration business, let me know, get in touch. And with this I'd be happy to take questions.
So, the next question, you start, also in German, I know that there are some German people around. So, I'm interested by the postdoc. No, I did already at my firm, so I will not come again. No, I'm joking. It was a very nice time,
Oh, really?
very nice place. Gregory was also at my firm, so we sat down too. So, first I have some questions, and some are really frank maybe. What is the minimal number of cells required to recreate a Planarian lab?
Very good question, Eric.
I know what I answered.
So, this is actually a relatively complex question, because when you think back about the pluripotency of the stem cells, and essentially, I should, explain it in a little bit more context here. So the way the pluripotency of the Planarian stem cells was demonstrated was by transplanting a single stem cell into an irradiated animal. And normally if Planarians are irradiated, clearly all the stem cells die, as in any other system. And Planarians will then consequently die over a course of about 3 to 4 weeks, simply because no new cells can be made. But in this case, transplantation of a single stem cell is enough to rescue the entire animal, which demonstrates that basically, again, the stem cells are pluripotent, and therefore one answer to your question is that in a way, a single cell is enough. But on the other hand, I think still, even if we would have the conditions to culture the stem cells in vitro, and we don't at the moment, but even if we wouldn't get an organism out of a single stem cell, because the stem cell requires the cues from the pre-existing tissue. And this was in fact what I was talking about in the second last slide. So I think there needs to be the definition of dorsal and ventral, and there needs to be the definition of this intrinsic tissue polarity in order to instruct the stem cell descendants what to do and where to go. And so basically these experiments have been done partially in the 50s and in the 60s, where the smallest that people could get down to were about 5,000 to 6,000 cells. But my gut feeling is that it is not so much the number of cells that's important, but rather the context. So whether you have a piece of dorsal epithelium and a piece of ventral epithelium. But yeah, we would like to test these questions in more detail.
And it looks like the beta-catenin is responsible of everything, basically. So there is tons of molecules, tons of clone, screen, and whatever, and there is coming only the beta-catenin, and there are also other molecules giving the same phenotype.
Yes. Yeah, I know I didn't go into this, but we basically get the same phenotype. We get a beta-catenin phenotype when we knock down all positive regulators of the pathway, so also some of the Wins give the beta-catenin phenotype and so on and so forth. And we also, if we target other negative regulators of the pathway, for example axin, then we also get double-tailed animals. So it's clearly, we see the phenotypes as we should be, the pathway is working the same way as it does in vertebrates. But what I find always amazing is that the knockdown of a single gene and the organism-wide knockdown of a single gene doesn't just result in a mass of anterior cells or a mass of posterior cells, but we get a perfectly patterned structure, only the opposite of what we would expect. So therefore I think what beta-catenin probably does is to act as a sort of selector switch very high up in the hierarchy of establishing cellular organizers, like in development. And just by switching beta-catenin, you switch the type of organizer you form from tail organizer to head organizer.
No question?
You said that your organism has 5 EFGR and it's quite rare at this level. Did you check where the duplication comes from, and is it shared with another type of organism capable of regeneration? Something that you went through? Or?
We haven't analyzed specifically other invertebrate genomes, so I can't really tell you at which point this duplication or multiplication happened. Just 2 points. So just reading general reviews, the consensus or the dogma is invertebrates generally have only one EGF receptor. And what we try to do specifically is to build phylogenetic trees between the 5 Planarian EGF receptors and the vertebrate, the 4 different vertebrate EGF receptors. And what we found is basically that the Planarian split of the family was an independent event. It had nothing to do with the 4 different EGF receptors in human. So it was an independent gene multiplication. But we actually see this quite a bit in Planarians. So for example, also with TGF-beta signaling, where they have lost some members, for example, cordin, which is really surprising, but they have massively amplified other gene families like noggins, for example, of which there are 10 in Planarians and only 3 in vertebrates.
Do you think that EGFR also involved in tumorigenesis?
Sorry, again?
Do you think that EGFR is also involved in tumorigenesis as you thought?
Right. And so that's why I stress so much that this EGF receptor 5 phenotype is specific to protonephridia, because the 4 other EGF receptors, they do exactly what you would expect an EGF receptor to do. So there's one of them that when it's knocked down, it clearly, basically stem cell division goes down massively, and the stem cells hardly divide anymore. So very likely this EGF receptor is in fact required for maintaining the mitogenicity of stem cells. And interestingly, there's also a second EGF receptor that has the opposite function. So when you knock it down, there's more stem cell division. And this is something that I find really interesting now in terms of this gene amplification in Planarians, because also in humans, of course, there are positive and negative acting EGF receptors. And so if Planarians have now independently diversified the EGF receptor family, and they still arrive at one that's positively acting on cell division and one that's negatively acting on cell division, you just wonder if there are any other topological constraints in the network that lead to, this, in this case, probably convergent evolution.
You were talking about dorsal and ventral, and the stem cells, the dorsal part and the ventral part are similar. It's exactly the same stem cells, or they have some different function, which can explain the fact that you recreate more the dorsal than the ventral in function of the curve.
So at the moment we have absolutely no idea about heterogeneity within the stem cell population. So therefore, I can't answer this conclusively, but the fact, again, that in Peter's hands, the transplantation of a single stem cell is sufficient to generate the entire animal would at least argue that, stem cells can make both dorsal descendants and ventral descendants. Now, whether they do within the normal environment of the animal is a different question.
Just one more. Sorry.
Eric,
I'm curious. You know that I'm curious with that because we are so much.
Yes.
Yeah, but I'm not sure that my liver will survive here. Postdoc in Germany. When you show this planaria with 2 tails, there is brain or no brain?
There's no brain.
And so there is no neuron also?
No, there are plenty of neurons. But
yeah.
So basically, in this case, it simply shows that Planarians don't need a brain simply to survive.
But is eating and
no, they are not eating.
So,
but on the other hand, what is still present are the ventral nerve cords and the nerve net. So basically, there are lots of peripheral nerves that surround the Planarian body, basically with a nerve net. And actually, this nerve net is capable of rather sophisticated computations. Because, for example, if you take a decapitated fragment, so a fragment without the brain, and you turn it on its back, it will always right itself again. So it can both sense the gravity vector and it can translate it into a writing response independent of the brain. And moreover, these brainless fragments can also find the darkest corner of the dish, for example. So they must have a way to sense light and to again integrate this into behavioral responses. So basically, the bottom line is that the Planarian nerve net is capable of sophisticated computations and can keep the animal perfectly alive. But probably what the brain is doing is to allow the sensory information incorporation for prey capture and for feeding.
And the neuron regeneration, it's working like for the organ, for the head, everything is the same system.
What do you mean?
If, when you cut the planaria, you cut the neuron when you amputate the planaria. And the regeneration of the neuron part, neuron organ, it's a similar system that for the rest of the organ, the rest of the organism.
These are really big questions at the moment, and the problem with neuronal regeneration is that so far we don't have the markers, to look specifically whether a specific subtype of neurons may be missing, and secondly, so far no one has found any RNAi phenotypes that block neurogenesis, for example.
No, just a question, because the capacity of regeneration is very important. So you think that at one point we can find something that we can use in human, for example, to help the regeneration of one organ. I don't say that we can regenerate everything or whatever, but just some molecules or mediator, or maybe a mix of stem cells and protein cells.
I would say very indirectly yes, and my fundamental approach to this problem, to this translational problem of regeneration research, is that I think what is fundamental to regeneration is to have patterning systems active at steady state. So meaning that if you do make new cells, they have one way of knowing what they should do. And I think one interesting view of cancer, for example, could be that this is in fact producing new cells without telling them what to do. And actually there's interesting data of, for example, when you take very, very malignant cancer stem cells or cancer cells that would kill a mouse if you transplant them. If you put them, the same cells, into a mouse embryo, into a developing embryo, they behave completely normally and perfectly normally and like integrate into tissues and just participate in development. And so again, I think what we can ultimately learn from planarians in terms of translational research is to understand what should be these patterning systems and how they should work in order to give rise to a self-organizing system. And then we can, think about reactivating such systems again, maybe also during human regeneration. But of course, planarians are a model for fundamental research. They are not a translational regeneration research model.
I agree with that. It's just a question, a way to see the things. Since we are at the hospital, not.
That's why I was stressing that protonef really are a good model for doing kidneys.
Ok, s'il n'y a pas d'autres questions, c'est bon? Ok, thank you very much, Ron.
Thank you.